Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today is Oliver Ryder, PhD. Well, he is the Kleberg Endowed Director of Conservation Genetics at the San Diego Zoo. And we're going to be talking about plans to build a genetic Noah's Ark that would have 66,000 plus species. So, Oliver, thanks for coming. How you doing?
I'm doing fine. Thanks, Richard.
Yeah, tell me a little bit about your work. How did this idea come about for this genetic ark?
Well, the impetus for it was really to use advancing technologies to aid in the conservation of endangered species. And it's only been about 60 or more years, around 60 years, that it's been possible to grow cells from an animal outside of the body, to grow them in the lab and freeze them and revive them so that they can carry out their function. And this provided immediately some advances, significant advances in scientific studies and in understanding the evolution of life, being able to study chromosomes and identify in humans and other species that errors in the transmission of chromosomes, errors that take place across generations, cause diseases and cause problems for individuals. So in that background of this technology, half a century ago, when the San Diego Zoo started its research effort focused on helping to conserve endangered species, the founder of that effort, a physician named Kurt Benerschke, brought with him from the medical school at the University of California this technology and started what he called the frozen zoo. And so over the years, this collection has grown to be probably the largest, most diverse, best characterized, and most utilized collection of its kind. And although it has all of those attributes and accolades, it contains the cells of 10,000 individual vertebrates, animals with backbones, so fishes, reptiles, amphibians, birds, and mammals. There's approximately 1,200 species, but there are over 60,000 species of vertebrates. Now we realize that there's never going to be a time when we could collect more samples, more biodiversity than now, because we're seeing an increase in the loss of species and the declines in their numbers. And also technology has arisen that having access to these kinds of living materials from animals can contribute substantively to studies that benefit their conservation.
What about the study of their attendant microbiomes, their viromes, fungi, all those things? I mean, just that are, associated with those species alone that you're cataloging. Then all the omics, you've got maybe genetic material, but I mean, I know it's a monstrous undertaking, but is there any thought to gathering that info afterwards?
Sure, we think about that. And you're right, it is a monstrous undertaking. And we hope that these efforts can expand, but certainly it would be a priority to expand the number of species for which we have viable cell cultures frozen. And so priority is to expand efforts so that other facilities like the ones that we have at San Diego Zoo Global exist in other countries and in other regions of the world, so that there would be a broader catalog of species that could be saved and or could be studied The microbiome, of course, is very important. We understand how that influences animals, and we are there's a call and there's an effort to have a global microbiome, but that's another complicated undertaking, and we're going to be happy to contribute to that. But probably our biggest contribution is going to be this bank of living cells. It includes also reproductive tissues and cells.
Okay, so you're not just taking the DNA, but you're taking the entire cell, and
Yes.
you're taking some of their gametes, their not the somatic cells, but their the cells that produce egg and sperm.
Yes, we have the living cells that we can replicate. The gametes need to be used for fertilization. They don't divide until they produce an animal. Or a zygote, and so this is a unified kind of effort, and we feel confident that this will benefit the future, and that as more is being learned and greater uses can be made of these cells to save species, it makes very good sense to collect more samples and bank more samples now.
Are there going to be a few anchor species that you're going to get a lot more data on than others?
Yes, for now, because we can, historically, we've collected samples from San Diego Zoo and the San Diego Zoo Safari Park and other zoological institutions. Hundreds of facilities have sent samples to us for us to bank. And but we've focused on cells that are high priority species conservation. So some of these are species that were extinct in the wild and were saved from extinction by zoos. So species like the Przewalski's horse or the Arabian oryx or the black-footed ferret or the California condor or the Hawaiian crow or alala, all of these are special targets. But now, increasingly, we see the need to integrate efforts to save species in natural habitats with those that are preserving wildlife in human care, like what zoos are doing, and integrating these efforts, because as species become more endangered, gene pools are shrinking to gene puddles, and that threatens the future prospects of a population, which for us underscores the need to really be doing more banking now.
Mm-hmm. So how far along is the project? How many samples do you have and how many more to go and over what time period?
Well, we have samples from 10,000 individual animals, and it's about 1,200 species and subspecies. It's richest in mammals. And among the mammals, it's richest in hoofed mammals and in primates and in carnivores. But it's got a broad genetic diversity. So the future is that, we want to continue to collect this kind of material, but probably our greatest impact can be to help foster the development of facilities around the world to share our techniques and to encourage others and to establish a global database about what samples have been saved from what species, so that we can keep track of this.
How will scientists get access to the database? Do they make a special request and pay a fee, and then, some of the cells are cloned and then given to scientists, or how will this be utilized?
Well, we do thaw the cells and expand them. So then it's a renewable resource. So it can be, but it can be greatly expanded. It can't be expanded infinitely because cells in tissue culture undergo aging or senescence. But by freezing cells when we have millions of them at early stages, and then managing the way that we expand those cell cultures, we can provide a great deal of material. And now it's possible for some species to make induced pluripotent stem cells from these kinds of cells, so that these are cells that can make any kind of cell in the body, and they are immortal. So we are working on developing that technology as well. We have a process that reviews applications. So scientists contact us and ask for access, and we are happy to, we receive those and applications and review them and try to fill as many as we can.
What have you learned in the gathering of the samples, things that you didn't know or were unexpected?
Well, the collection of samples in the frozen zoo has helped really inform several big projects. One is the tree of life, the relationships between animals using whole genome sequencing, and cells such as we have banked and in the frozen zoo, and tissues that we can bank from animals as the time we're getting some of the samples from the frozen zoo can be used for producing very high quality genome sequences. And by comparing the genome sequences, great insights have been gained into evolutionary relationships. For example, the understanding that whales and are derived from or have a common ancestor with cattle or hippopotamuses, and that the elephant is related to the manatee and to creatures to which it has very little similarity now, but we can trace their ancestry through DNA. So that's really informed the tree of our knowledge of the tree of life. In other cases, being able to produce these genomes allows us to understand something about the extinction risk of animals. And this is a very active area of research right now, how the small population effects have produced accumulation of deleterious mutations. And so we can identify, and also to identify the genetic heritable diseases, genetic diseases in endangered species, so that these can be better understood and managed and used to provide better health care and prevent extinctions.
How many of these species have not been sequenced before? Are most of them novel?
Yes, I think that, right from the very beginning, after the human genome was sequenced, the genomics community realized that to really reap the benefits of having produced a human genome, that had to be placed in the context of the species with which humans are related, to which humans are related. So originally there were 29 other mammals besides humans that were sequenced, and the frozen zoo helped contribute samples to those. And then there was a project called the Genome 10K to sequence the genomes of 10,000 vertebrate animals. And there have been now about 300 or 400, the count keeps going up, mammals that are sequenced. And you might think that that's a long way from 10,000, but we see how rapidly the technology is developing and its utility. And so the idea now is to sequence all of the 60 to 70,000 vertebrate species in a project that's Genome 10K is morphed into a vertebrate genome project.
Yeah, you can see correlations. You could further elucidate, lineage. You could look for the appearance of endogenized viruses and track them. You should be able to see a lot once you get, a whole bunch of these sequences and analyze them.
Right, and there's a work that's going on that's about to be shared through scientific publications that has to do with taking several hundred, 247, I think it is, mammal species whose genomes have been elucidated, and lining them up to see what features are in common and what features are different across the tree of life of the placental mammals. And that's providing very interesting insights into diseases, human disease risk or human disease assessment, but also to conservation activities, to learn more about populations of endangered species, once you have a reference genome, then to be able to do studies of more individuals and get a kind of an understanding of this genetic structure of populations and the interaction, the migration and between isolated population fragments and whether which subpopulations harbor more genetic diversity.
Well, because you're helping to run the project, when a lab wants to use the data, are they agreeing also to share the results with you? Like, will you be privy to see all the research that comes of this, first?
Well, we are we're participating in the broad call of many scientists to make this data immediately available to the larger community. So we are part of consortia that are committed to sharing this kind of information rapidly, because there's much more information there than any one team can analyze, and it's going to benefit broad segments of society. So it needs to be out there and shared, and that's one of our commitments.
But what if you did a series of regular publications, almost like meta-analyses of everything that's come out of your data? This year this is what's come out. This year this is what's come out, or every 6 months or so. It might be nice for for you guys to do a publication based on what's being used and how and what's being looked into.
Well, we'd love to track that, but it turns out that it's not very easy, because people may publish the data and not say where they got the sample from, and they may publish the data and we don't know about it. So that's but there's no doubt that we can track through, for example, GenBank. There's thousands of hits for San Diego Zoo as a source of samples, and there's hundreds of species that the DNA that provided for a study came from us. So I think it's sort of remarkable that a nonprofit zoo has contributed so fundamentally to this explosion of knowledge in the field of genomics.
Yeah. It'll be interesting if you, when you're done, or even now, you create a virtual zoo. As I look through the samples, let's say, a video comes up of the animal itself, and I can literally see, oh wow, the sequences for a black-footed ferret. Look at it. That might be another level of data that's missing if you don't have that.
Well, we're trying. I mean, that's we're thinking about, and that's sort of a backfilling kind of exercise. And there are other databases that keep track of animals, and at the global scale, integrating all of these heterogeneous databases becomes one of the big data opportunities and challenges of our time.
Yeah. I just think that'd be really cool to have, literally, because you're a zoo, do also a virtual zoo like that, where you could look through things. So very cool.
We and other people are working on things like that.
Yeah. What's your estimated time for it to get to the next 10,000 and then to get to all 66,000?
I think it's going to take a while. If you look and find out, for all of these named species, some of them, a significant number of them have, were collected very few times. And if you went out to try to find them, it could take a long time. It'd be prohibitively expensive. So the idea is to sort of start on the things that are the easiest to get a hold of and encourage, efforts. But, all around the world today, there are wildlife scientists and forensic biologists and law enforcement officers, people who have their hands on animals that could collect a sample. And if there was a network in place that was ready to receive samples from rare opportunities to obtain a specimen from an animal, then we could make a lot more progress. I think that the way that we've been building the frozen zoo is adding 300 to 400 new species a year. So that's going to take a long time to get to 60,000. That's why we need to have more people involved. And it'll be very tough for fishes because some fishes have been collected only rarely. And the ocean is a big place.
How many of each creature or species or subspecies is enough to have? How many different examples?
Well, that's a great question. And, we would like to have, more is better, basically, but we have a limited capacity to process samples. So we look at something around, 20 to 50 individuals. It depends if they're related, and it depends if it's a species of that we're really focused on. For some species where we have basically the entire gene pool of a species, we want to be sure that we collect that. For species where we have the opportunity to collect the entire gene pool, we should be able to do that. For other species, we might want to have a representative examples. But, if we want to use these samples for genetic rescue, we want to be sure that we could reconstitute as much as possible of the genetic diversity of a species that's on the brink of extinction.
Yeah, this seems like a very important fundraising type thing. I don't know if you're doing this, but I would say, like, if you put out a call to action, we need funding, and, if you pay for sequencing of a particular creature, then you get some kind of, name recognition or bonus. And maybe people would like that if they could say, you know what, I really want to sequence dolphins. And so they fund you and you go after dolphins, and someone else likes ferrets or, whales or whatever it is. That might be a nice way to fundraise and get people really into it for particular types.
I think that's a good suggestion. And I think that we want to try to have this become more a process of global ownership and a broad recognition of the importance of undertaking this kind of work and having supporters for it. So we do have a wildlife conservancy that people can donate to, and a portion of that money goes to efforts that we use to save particular species, and that includes banking specimens from that. And when we look at our conservation projects, we look at the full spectrum of conservation, preserving habitat for the species, mitigating against the causes of its decline, but also trying to bank specimens so that we would have important resources for contributing to the recovery of species that are in decline.
Are there any particular ones that, it would be easy to get funding for, and other ones maybe that aren't cute or weird or scary that no one really cares about that would be difficult to get funding for?
Well, it's really complicated, Richard. Many people love tigers, and so we can get tigers from zoos, and many people love rhinos, and we can get rhinos from zoos, but there's one species of rhino, the Javan rhino, that none of them are in zoos, and nobody has ever collected a sample from them. There's no cell cultures from Javan rhinos. And people are very attracted to and allured to the great apes, but like gorillas, but we don't want to harm them or harass them. So how can we get the samples from gorillas in the wild? That's really precluded at this point. So the targets of opportunity have an interface between scientific interest, popular interest, ethical considerations, which are a huge component of our work, and then being able to export those samples to a laboratory. Numerous details.
Well, very good. What's the best way people can find out more and donate and see what's left on the list to sequence?
Well, thanks. We have a website that's institute.sandiegozoo.org, and there you can find out about our conservation effort and look to see about our frozen zoo and other projects we have for conserving endangered species.
Well, very good, Oliver. Thank you for coming on the podcast, and what you're doing is really important. So I hope that you guys get some real kick-ass marketing and get these sequences going, and as soon as possible. So I'm glad you're doing what you're doing.
Thanks for your interest, Richard.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.